Nós estamos aproveitando a visita dos professores Hans e Carsten, e o tema que vocês estão vendo já no próprio título da palestra, né? Então nós estamos estudando aqui, investigando a parte de isótopos estáveis, composto específico, né, para caracterização de degradação, biodegradação principalmente em situ. E nessa semana de visita e início de trabalhos, nós estamos aproveitando então a presença deles e solicitamos então uma palestra na sequência. Okay? Então nós temos a satisfação de ter a presença e eu gostaria de agradecer ao professor Ramos e ao professor Castro, né, pela disponibilidade. Então essa é a nossa sequência.
Thanks for coming for our seminar. So today here I will talk about here compound-specific isotope fractionation analysis for characterizing in situ degradation. As you know, we have heard a minute ago, we will have a common project, and in this common project, which is funded by Petrobras, we will use this technique, isotope fractionation technique, to characterize in situ degradation. And here, for today, we try to here shape the seminar a little bit like that I will make an introduction, and here then I focus on this isotope fractionation technique, which might be to some extent also new for you. And Carsten will talk about complementary techniques, how to characterize in situ biodegradation. So here I start with the acknowledgement. This is our team, yeah. So that was our team in 2013. So about a little bit more than 35 people which are working in our department, which is called Department of Isotope Biogeochemistry. It's an interdisciplinary department which has geologists, microbiologists, chemists, and physicists. Yeah? And we need all these expertise to characterize, to develop clear concept to characterize the microbial processes, soil, aquifers, or sediments. We have funding from here, different funding agencies, for example, European, German funding agencies, German Research Foundation, State of Saxony, and others. So we are doing a lot of third-party funding, obviously. But now coming a little bit here to the topic today. This gives an overview of different techniques which we are using for the assessment of microbial degradation process in the environment. So this is a typical contamination scenario where we have here leakage of a contaminant to a soil aquifer system, and we were particularly interested in natural attenuation to characterize it and quantify this. And therefore we use complementary techniques. Not only one, we're looking at this subject from various angles. So we are doing field work, we are developing test systems, Carsten will talk about that. We are using molecular biology, like gene markers. We are doing enrichment cultures, so taking material from the environment and trying to isolate and characterize the microbes which are doing biodegradation, and we have different techniques like stable isotope probing here for proteomics or from DNA, so molecular biological tools to analyze the structure and function of microbial communities. And that is where we are interested in. We are interested in the structure, function, and activity of microbial communities which are governing natural attenuation or in situ biodegradation process in the environment. We want to analyze factors which are controlling the microbial activities. So find out some limitations. When we know the limitations, we think we can manipulate the system also to enhance a little bit biodegradation. So that it comes a little bit in the direction of biotechnology. We want to have process understanding for managing contaminated aquifers. That is in particular important for natural attenuation concepts. And we want to use these techniques for control of remediation measures, if these are successful or not. So this is the overall questions where we are concerned with in our department. And I'm talking today mainly about the isotope fractionation concepts and the geochemistry. So, the isotope concepts for assessment of microbial degradation in aquifers are important for several subjects. So, for example, for risk assessment, we want to know what is the risk of that. So here, knowing the half-life times or the persistence of contaminants, this is some criteria which is here needed to assess the risk. We want to develop remediation strategies, as I mentioned, evaluation of remediation measures, and we want to develop management strategies for contaminated aquifers, in particular the natural attenuation concept. I will come to that point back here a little bit later. So natural attenuation is these processes which are leading to a reduction of the contaminant mass in a given environment or in an aquifer. So attenuating the concentration, the actual concentration. And there are several processes. And the US Environmental Protection Agency the US Environmental Protection Agency has established a so-called three-tiered approach to implement natural attenuation. And for this three-tiered approach, three main questions have to be answered. So one is demonstration of, for example, decreasing contaminant mass over time, for example, as at a monitoring well or within an aquifer plume. The second one is indirect demonstration of natural attenuation processes active at the site using hydrological and geochemical data. I mean that you are also doing when you are analyzing when you are analyzing electron acceptors, for example, and other geochemical conditions, so change of alkalinity or things like that, but these are indirect indirect tests. So consumption of electron acceptors might be reflecting the degradation of contaminants. So that would be like an indirect approach. And very important, direct demonstration of a biological degradation process at a site. So this is also one of the key questions would need to be addressed. And looking at a contaminated site scenario, we can think about what are the factors, what are the processes which are affecting concentration there, and what are affecting the reduction of mass of contaminants. So when we have, let's say, a scenario like a leaking plume, so we have concentration gradients which are controlled maybe by absorption to the aquifer matrix. Then it might be hard to analyze that. So it might reduce the concentration in the water which are forming the plume, but do not change the mass of contaminants. So absorption might attenuate the concentration in the groundwater, but do not change mass of contaminants. Also volatilization, which might affect concentration, is only a transfer of contaminants from one compartment to another compartment, but do not change the mass of contaminants. As well as dispersion and dilution by bypassing water. So when the plume is expanding and mixing with pristine water, you are expecting dispersion and dilution, which affects the concentration but not the mass of contaminants. It's also only a dilution. And in particularly in Germany or in Europe, for the natural attenuation, we need to demonstrate that there is a removal of the mass of contaminants. And this is, for example, mineralization, biodegradation of organic contaminants to CO2 or to methane, depending on the given geochemical conditions. And here, therefore, in particularly in Germany, it's very important to demonstrate biodegradation, because this is the only sustainable process which are reducing the mass of contaminants. And that is what we are going to do with stable isotope techniques. So now I'm going a little bit back here to the fundamentals. So organic contaminants are consisting of carbon, hydrogen, to some extent of nitrogen and oxygen. Those elements have at least two isotopes. Here, oxygen has 3 stable isotopes which are not here, not reactive. Yeah? So the heavier isotope is appearing in that large higher masses here as atom percentage compared to the lighter isotope is appearing in much higher here natural abundance compared to the more heavier isotope. The ratio of light isotope to heavy isotope we call isotope ratio. And this isotope ratio is reported in delta notation relative to an international standard. And for carbon, this is the PD belemite. This is a rostrum of a creaceous fossil from the PD formation. So this is just this is here the way how isotopes are reported. So like a convention. And this is reported in per mil deviation to the standard. So when we look at the natural abundance, we see that here there are slight slight difference. So 1.1% it's here is the natural abundance of 13C for carbon. For hydrogen, the natural abundance of deuterium is much lower, and this is here for nitrogen, oxygen, and sulfur. And we want to make use of the isotope ratio as some of the process parameter. And here, only in this decimal, there is a change of the isotope composition due to natural processes. And this is where the information is. So, for measuring very small relative amounts of your heavy to light isotopes, so isotope ratio, we need sensitive instruments. An instrument, this is an isotope ratio mass spectrometer, here connected to the GC. And here the uncertainty of this technique is approximately 0.5 per mil. And this is so what you almost expect as a natural variation. Yeah, so these natural variation we want to measure with these systems here as a process parameter, for example, for characterizing biogeochemical processes, in particular here in situ biodegradation. And this is approximately the uncertainty. And this is the variability for carbon, which you approximately will expect. And when it goes to more positive values, we say that this is enrichment of the heavier isotope, where when it's going here to more negative values, then it's a depletion. So then a depletion of the heavier isotope, so an enrichment of the lighter isotope. So that you need to know for the nomenclature. So the system which we are using is GCMS, which is connected to an isotope ratio mass spectrometer. Here, showing the gas chromatograph. In the gas chromatograph, we are separating mixtures of components to individual signals, peaks, which are then oxidized in a furnace to CO2. And this CO2 is then subjected to an ion source and registered here by a detector system, and the light isotope is quantified compared to the more heavier isotope. And we use the standard with known isotope composition as a reference, yeah, where we compare our signal from our sample with the standard. So this is the way how we are doing it. You see already here, this is different than the GCMS system. For a GCMS system, you can use selective ions or something like that, but here you need baseline separation always, which is sometimes quite difficult to achieve when you have very compact mixtures. And this is different to normal GCMS systems. And on top of that, this system is also not so sensitive like a normal GCMS system, of which some of you are probably sometimes knowing this. So we have different isotope effects. So one well-known here isotope effect is the so-called thermodynamic effect. For example, for evaporation of water. Yeah, so this is like a distillation process and accumulation in clouds. Here, with this evaporation, you have an isotope fractionation factor about this here at 25 degrees. So this is like a distillation process. And for contaminants, also volatilization of components out of a water phase. This is not a distillation process, this is a phase partitioning process. So it's a phase partitioning between the air and the water of a different chemical, for example, trichloroethylene or dichloroethylene, which are partitioning between the water and the air phase. And this is also associated sometimes with an isotope fractionation factor, but this is also very small. This is a very, very small isotope fractionation process. And it characterizes the abundance in different phases, so in the water or in the air phase, or here the distillation of water molecules out of the water, here like evaporation. So evaporation is here, the evaporation and the water cycle has been characterized since many years using isotopes. So by that here, the evaporation is calculated, the isotope effects of the evaporation is calculated, and you have the formation of clouds when water is precipitating out of that here. here the oxygen and the hydrogen isotopes in the water phase are heavier than here in the gas phase. So when it's raining out, the cloud will become isotopically lighter and lighter, and that can be used, this information can be used here to characterize the water cycle. For example, at high altitudes, you have very light water, or at high altitudes in cold here environments, you have very light water. Or, for example, you would find very light snow on top of mountains or something like that. So that is used for the water cycle. So, and these are thermodynamic isotope effects. In contrast to isotope fractionation processes, which are related to kinetic isotope effects, outlined here. So the principle of these fractionation is different. This is not a distillation process or something like that, but this is a bond change process. So here, this is a kinetic isotope effect, which is here related to a cleavage, a chemical cleavage of a bond. A biochemical cleavage can also be a chemical cleavage, which is separating isotopes. And here, the principle of that is that here, bonds which are made up by light isotopes are faster cleaved than bonds which are made up by heavy substituents. I'm coming here to that a little bit later. And here, the consequence of that is that the transformation products are isotopically lighter, and the residual fraction become isotopically heavier. This is known for natural processes like methanogenesis, methane oxidation, and we explore that for the degradation of contaminants since almost 15 years. The principle of the primary kinetic isotope effect is the bond cleavage in the transition state. And this is here a scheme of 0-point energy which is needed to activate a bond that you have here, a bond cleavage. And bonds which are made up by heavy isotopes, here for example deuterium compared to the hydrogen, require a higher activation energy than here bonds which are substituted by hydrogen. This is very simply due to the factor you have here an elastic vibrator, and here when you have here when you have lighter isotopes, the vibration can be larger than having here heavier isotopes. And you need a certain vibration and a rupture of bonds. Yeah? And therefore, the rupture of bonds with hydrogen is faster than the rupture of bonds which are made out by deuterium. And this is a very fundamental process. Can be modeled by the Arrhenius equation and characterize the bond cleavage of the transition state. So it reflects already type of mechanism which is leading to this bond cleavage. That we should remember, because we want to make here use of this. So the effect of this is outlined in this cartoon. Yeah. So for the bond scission, yeah. So lighter bonds will react faster than heavier bonds. Yeah. So here we have a mixture of isotopically light gray and isotopically heavier components. And here, when the lighter is reacting faster, then those isotopes we will found in the degradation products, and here in the remaining fraction, which has not reacted yet, we will have an accumulation of the more heavier bonds. So easy to remember. And that can be modeled here by this generic example. This is here the fraction of reacted. Yeah? So here our reaction starts. And when we always transform the vital components a little bit faster, this is here the isotope. This curve reflects the isotope composition of the Isotope or the reactor. So it will become heavier and heavier. And by this effect, yeah, this is our fractionation factor. First we form very light product, and in the closed system that would becomes heavier and heavier. This is the accumulated product. And when the reaction is over, the product should have, for mass balance reason, the same isotope composition as the reactant at the start of them. And these changes are simply what we are trying to interpret in the environment. So we want to make use out of these changes to interpret the process in the environment. And that here we will explore in the next half hour. So, an isotope fractionation process is first studied with reference experiment. This is a reference experiment in a culture where the only sink of contaminant is biodegradation, because we want to have the isotope fractionation factor for biodegradation. So this is the experiment. So in the degradation experiment, toluene with the hydrocarbon is degraded. Here we have the isotope composition of toluene here in red. It becomes heavier in the residual fraction by several delta units. Sulfide is formed because this is a sulfidogenic culture. And here we want to quantify the change of relation, the change of here isotope signatures between beginning and after a certain time of reaction, and correlates this with the change of your concentration. And this is correlated by the isotope fractionation factor alpha. Graphically, you do it very simple. You are plotting here the logarithm of change of concentration versus the logarithm of change of isotope composition. The straight line gives the fractionation factor and quantify the fractionation process. Here, the calculation which we are used for that is the so-called Rayleigh equation for distillation, which relate here the change of the isotope composition to the change of concentration. And these fractionation factor, we need them when we want to interpret our change of isotopes in open system like here. So, in the beginning we were very interested to characterize here different mechanisms. So, as we heard, so the mode of bond rupture has a certain specific isotope effect. So we investigated this very systematically for different biogeochemical conditions. And this is an example for the degradation of toluene. From the literature, there are different modes of bond cleavage. Yeah? So there is, first of all, dihydroxylation, which is using molecular oxygen for aerobic degradation, which introduced here two oxygen, added this to the aromatic ring. A monooxygenation, which add here OH to the aromatic ring. Monohydroxylation of the methyl group, which is obviously a different mode of bond cleavage, reacting here molecular oxygen with the methyl group. And under anaerobic condition, this is here the so-called benzylsuccinate pathway, which activates the toluene by adding succinate to the methyl. So quite different here mode of bond cleavage of here of a carbon hydrogen bond as a methyl group, or at the carbon-hydrogen bond at the aromatic ring. And now we want to see if we can check this using this isotope fractionation. And what we need to consider there, so these are the different points of reaction at the toluene here molecule. And in biological systems, we would need an uptake of here the molecule to the cell, yeah, which might be some kinetic step, but it's not isotope sensitive. We might have a binding of the substrate to the here enzyme, which is reacting. The binding is like a phase partitioning. So maybe there is an isotope process step associated with it, but it's expected to be very small. And then we have the isotope very sensitive step, and this is the bond cleavage which makes the reaction irreversible. And so we are investigating this now systematically for different reaction and here at different, which are reacting with different here position of the toluene molecule. And we have done this, we have analyzed the carbon and the hydrogen isotope fractionation of here a number of reference reaction with all these organisms. So starting with Rhodococcus or here, which has a carbon isotope fractionation. Here, these are the changes upon reaction of here the carbon isotopes, where we plot here the hydrogen isotopes, and then we see there is here almost no or even inverse fractionation associated with the biodegradation here for hydrogen, but there is some for carbon. And another strain, Pseudomonas putida, MT2, this is this curve. This has a strong carbon fractionation and a very strong hydrogen isotope fractionation. From biological, here, previous biological reaction, we know that here, Pseudomonas putida, this here is reacting with a methyl group, yeah, so activating toluene as a methyl group, while here this Rhodococcus strain is reacting the toluene at the aromatic ring. And that gives a very different fractionation pattern, even when the same electron acceptor, in this case oxygen, is used. Yeah? And here the others are sulfate reducing strain, or here strains which are using here nitrogen as electron acceptor, and these are falling in these categories and in these categories. In summary, when we are using here carbon and hydrogen isotope fractionation, we have something to distinct different pathways according to different electron acceptors, as when we are using hydrogen and carbon isotope fractionation. And that would allow us maybe next to quantification, that maybe also allow us to analyze, dominate reaction pathways also in the environment when we want to characterize our degradation process in the environment. And that would be very difficult to do that with other methods. So what we have learned until now, physical processes leading to very small fractionation, but biodegradation would do. So when we have change in isotope composition, we would, in the environment, and the only likely process is biodegradation, then we can use this changing, this change enrichment of isotope composition to track biodegradation. It seems to be logical. And this is a concept which we have developed. So what we are doing, this is a scheme of a field site. You have their concentration. We are monitoring them next to concentration. The isotope compositions. We are selecting a compound-specific isotope fractionation factor from reference experiments. We're using the Rayleigh equation distillation for low, high, or intermediate fractionation to correlate changes in isotope composition to here degradation, and then we are monitoring here down gradient here. We try to monitor the degradation down gradient here, a source of contaminants, for example. So, what possibilities for degradation we have to characterize? This is again the Rayleigh equation, where the compound-specific isotope fractionation factor alpha is correlating change in isotope composition to change in concentration. So we can use then in the environment here the change here of the isotope composition between a source and a monitoring point, for example, selecting a specific isotope fractionation factor to quantify or characterize biodegradation as percent, and we can relate this to the initial concentration for a flow path, or we use here the concentration at a specific monitoring point to simulate what might be the initial concentration in the flow path which is going down the drain, and what might be here the degradation between a monitoring point and a hypothetical here entry point of contaminants to the environment. So these are some of the options you have. So we have published also a paper where this is more put here to real modeling approaches to numerical models. And this is here published by Tullner in 2009. But these are this shows this is a principal use of the Rayleigh equation to characterize the degradation, making use of the isotope fractionation. And this is one of our very old examples. This is an aquifer in Hamburg. So there were erratic mines where underlying this is sand aquifer to some extent. And here it was covered also by erratic mines leading to confined conditions. We have measured here the isotope composition of benzene here in well A, B, C, D, E, and F. This is here shown the measured isotope, here the measured concentration, down gradient here. This is the isotope composition, and we have used here the Rayleigh equation and a specific isotope fractionation factor for sulfate reducing condition, because it was an anaerobic aquifer, to calculate the concentration of toluene, for example, down gradient of this flow path. And the calculated and measured concentration not always match, but in principle it match. So we conclude that biodegradation is a major thing. That would be the approach. So a more complex here site is our experimental site in Zeiss. This is a former hydrogenation plant located in the vicinity of Leipzig. The other example was here somewhere. So this is a very strong benzene contamination going close to solubility. So in the source zone it's higher than a gram per liter, and the plume is stretching down here more than a kilometer. We are monitoring the concentration of benzene in between A source zone and then different monitoring wells. And again, the question was, what is a major degradation process? And also for benzene, there are a number of different here potential degradation reaction. So with oxygen, you have here monooxygenation or dihydroxylation, and here anaerobic degradation is not so clear, maybe carboxylation or other reactions. So we are investigate this systematically with defined cultures. And again, we see here the systematic of benzene degradation. Here is the change in carbon isotopes, and here is the change in hydrogen isotopes. And for dihydroxylation reaction, again, we find with a number of reference experiments only very slight hydrogen isotope fractionation, where some significant carbon isotope fractionation. So when you find this at the field site, there is no hydrogen fractionation, but carbon isotope fractionation, you can conclude that benzene is, for example, degraded by an anaerobic process. So monohydroxylation reaction gives some hydrogen fractionation and strong carbon isotope fractionation, and the anaerobic degradation pathways clustered here, having strong carbon and strong hydrogen fractionation. So you can see already from this graph, you have some diagnostics when you use these concepts. And then we have done here analyzing here the fractionation pattern down gradient to source. So this is a depth-related sampling. The water table was approximately at 10 meters below here surface, and here this is the shape of the plume, this is the concentration. So the highest concentration of this plume is between 14 and 16 meters, and here the carbon in red and the hydrogen isotope composition is inversely correlated with here with the concentration, which might show already that at the fringes, upper and lower fringes, there is more degradation than in the center of the plume. This is the isotope composition of this for hydrogen and carbon for the source signal. So what we already see from this, we have a proof for biodegradation of benzene on the flow path here, whereas here at the fringes is a little bit higher degradation compared to the center, but still in the center is also biodegradation. So that is a way how you are analyzing your data when you have that. And then you can use the two-dimensional the correlation of carbon, your hydrogen isotope fractionation to analyze the dominant pathway. And here we are plotting all the data which we have from this field site, from this monitoring profile, which is also depth-related setting. And the development for carbon and hydrogen, change in hydrogen composition is going in that way. And this is the area for dehydroxylation and monohydroxylation, but the development was in this area, so it is clear that it is an anaerobic process. And here, of course, also the aquifer is anaerobic. You may say this is trivial, but I will show you an example a little bit later on where we do a consulting operation, and you will see this might be really useful to have. So in this case, it's anaerobic, analyzed by isotope fractionation. And then we can use our Rayleigh equation here, select a specific fractionation factor for your anaerobic degradation, and then we can calculate here the amount of biodegradation which we have. This is the source zone, and these are the different monitor reports of depth-related sampling on the profile down between A and monitoring well E. Yeah? And then you can process this like percentage of biodegradation, for example. Or you use here a mapping of that. You are comparing the differences here between source and here fringe of the plume, and you use again fractionation factors which you are taking from literature, and here then you can calculate here the percentage of biodegradation as a map. Yeah? You can look for uncertainties taking different fractionation factors according to your hypothesis. What is the different major degradation pathway or the dominant one, and you can use here the uncertainty which is related to the variability of isotope composition in the source. Or you can do other here calculation for uncertainty of your measurement, for example. And that we have done here. And this is for example a typical program. So in summary, today, after I think we are working almost 50 years, yeah, we have here for different pollutants group like PTEX, we have talked about that, but it's also working for PAH, here chlorinated hydrocarbon, fuel additives like MTBE, and it's working for plastics. So for these components here, a number of enrichment factors for calculations are there. Not so much for chlorine. This is still here a field under investigation. We are developing this. It's very much needed that you know a little bit on the redox condition when you're interpreting that. Today we have more than here 30 references available also for different field sites. So there are different here field sites explaining how to use this, and from year to year there are coming new examples how this can be implemented, also what is the uncertainty associated with this technique. We have found a company which have done a lot of here studies at different here contaminants, for example in Germany. So chlorine chemistry, refinery, gas works, tar oil or hydrogenation plants are typical here contaminations occurring in Germany, where we have done consulting operations using this isotope techniques. So it has been recommended by guidelines. There are EPA guidelines. Colleagues of mine has been here working for this. I was one of the review of these guidelines, for example. So there you can find more information. So, and I have some minutes more. So now I want to show you a field site which is maybe a little bit different, similar to your field site of the refinery. This is a field site in Weisland-Gölzau. That was a hydrogenation plant and a processing plant. They also do here some raffination of your tar oil and things like that. So the contamination was a local aquifer which is loaded with crude oil and also raffination products related to oil, PTEX, PIHs and phenols. So the concentration was we're leaching in this direction. And here, because that was in a field site where they do chemical factories, and they had a dome-like structure, and under the dome-like structure was made out of erratic miles, closing the aquifer to confined conditions. And here, below that was a very severe oil and PTEX contamination, which were related to methane. So methane was accumulating under a dome-like structure. So when you drill there, methane was blowing out, and they recognized it because one of the workers were smoking, And then you have a flame, he loses all of his hair, eyebrows and things like that. Get a little bit burned, not really severe injured, but here the flame was, from this drilling, was burning for some hours. So they say, this is impossible when we have a chemical factory here. And they want to see, so what are the criteria for methanogenesis? How is this related to biodegradation? Yeah, so and how is this related to the in situ degradation, which is, in this case, a good thing because it prevents the stretching of the plume. So it was a complex here, yeah, consulting operation. And we tried to do that. And the question was, is methane acceptable there? Because it poses risk. And how is this related to monitored natural attenuation as a possible remediation strategy? Because we cannot also not here dismantle here the field site to dig it out, because there was active industry there. So it should be something like in situ degradation. At least that was taken under consideration. So the main problem there for forming large plumes were benzene, not oil. The oil was there in the source area to some extent, but it was not moving, yeah, with a bypassing groundwater, because it's not so soluble. So forming hotspots may be used as a carbon source for forming methane, but the oil was not forming long plumes. But benzene and PTEX components to some extent, phenols were good water soluble, and they form the long plumes. And this is the shape of the plume. So that was the contamination. Here we have a long plume, and even here was very high concentration to some extent. The plume was not very strong. So, and then they asked us if we can characterize this field. So, what we do, we measure the concentration of benzene and correlate this with the isotope composition. And as you see here, down gradient, we found an enrichment of benzene, which is probably related to biodegradation of benzene. So we already see biodegradation hypothetically here attenuate this plume. So it was known that in this area very strong methane was, there was very strong tendency for methanogenesis, and here a slower tendency for methanogenesis. So here we measured the methane concentration. Here, that was the longer PTEX plume, and here was the major methane plume, both are stretching down. And we are characterizing here the methanogenesis, making use of isotopes. And we found significant pattern of methane isotopes which are characterizing acidogenesis. So this is a process which are fermenting the contaminants, forming CO2 and acetate, and then forming methane. So by using these isotopes here, we saw that here is a very strong signal for acidogenesis. Here the isotopes are particularly a little bit lighter, which is going to the tendency you have a little bit more here CO2 reduction. So methanogenesis was proven there as a very active process. And the methane which is formed there was a biological methane. So we can use the concentration of the methane directly and correlate this to the degradation of contaminants. And maybe also for the oil refinery, that might be also interesting. And then we look, but what's happened more downstream? And here we see that here the isotope composition of methane becomes lighter and more heavier. So similar as organic contaminants, it becomes fractionated. And we interpret this as very strong degradation of methane in same areas where we have degradation of contaminants. So we conclude that here the degradation of methane as a plume on one hand is not competing with the degradation of benzene, for example, as a major contaminant of concern from this investigation. So methanogenesis there was proven as an active process of contaminant degradation and even do not interfere with contaminant degradation further downstream where they may compete for electron acceptors for degradation. So we are looking for the concentration of sulfate. Sulfate was here a major a major electron acceptor which are driving here biodegradation. And here you see the concentration. We had 10 millimolar of here sulfate as a natural background in this aquifer. It's a sulfate-rich aquifer, and where the major plume of the contaminants are, we see here a depletion of sulfate. So sulfate reduction, we can conclude from that, is an active process which are leading here to degradation as an indirect. And we combine that with isotope fractionation. So here the sulfate, which is a natural, is the natural background is between plus 0.2 to plus 0.6 or plus 0.1 delta per mil. In the course of the plume, it becomes very, very heavy. So this is shown the active sulfate reduction by the microbial community. So this is a way how to characterize these processes, making use of isotopes a little bit better. So, and we see that in this part, the plume, benzene plume, was very, very short. Here it was a little bit longer, even become degraded, but we conclude there that here possibly methanogenesis and strong sulfate reduction are some of the major biogeochemical processes which are removing here the benzene from this plume. So we also tried to calculate here the amount of sulfate reduction correlating the change of concentration of sulfate to isotope enrichment of sulfate, and this is clearly correlated, indicating this is a biological process. Another thing which was interesting was here the concentration of iron. Yeah, so iron, you have here laterites, you would expect a lot of iron. So iron mobilization or iron depletion is some indicator for microbial process. But what you will do when you have sulfite reduction, when you are forming sulfide, your sulfide will reacting with the iron, precipitating. So only the iron concentration by itself, it would be difficult to interpret that, at least in a quantitative sense. And what we find here in the center of here the plume, where we have indication for sulfate, strong sulfate reduction, so formation of sulfides, we have also depletion of iron. So that would not mean that iron reduction may not be present here, yeah, but it means that sulfide is precipitating. Yeah? So to characterize these processes in total a little bit better, you need to correlate all those things. But here we have also some iron mobilization, which is my point that iron reduction is also an important process. So we made a conceptual model for the environmental agency, summarizing all this data. So we have methanogenesis, we have here in the center of the plume. The plume is actually very nicely reflected by the concentration and isotope composition of the CO2 or the DIC, which might be also useful to do. So methanogenesis we have in the center. So we have methane oxidation down gradient the plume. We have high sulfate reduction in the northern center of the plume, and that is a little bit slower in the southern part of the plume, where we have a little bit lower sulfate reduction. And here we have an area where we have here maybe lower microbial activity without a clear dominance of terminal electron acceptance. If it's methanogenesis, it's iron rejection, or if this is sulfate reduction. So we want to characterize this a little bit better related to the isotope fractionation of the benzene. And here we have correlated the ratio of benzene down gradient between source and down gradient the plume. For hydrogen, these are the hydrogen isotope composition is the hydrogen isotope ratio, and this is the carbon isotope ratio. Here, the solid line shows one example for low isotope fractionation of hydrogen related to aerobic process and higher isotope fractionation to anaerobic process. And similar, the probability fields for carbon isotope fractionation. Not all these points are just fitting to our model, but the tendency show that, and here those points which are lying only on this line show no effect on the isotope composition, so might be some effect on the concentration, so we conclude this is mainly dilution. So we can separate reactivity from only dilution. And here then we, at the final step, we use again our scheme for interpreting the changes of carbon and hydrogen isotope composition. We plot here the field data. So there are some areas which will fit to aerobic degradation with oxygen, for example. There are fitting some to iron reduction here, and the others are fitting to maybe methanogenesis or unknown degradation pathways, which showing very strong hydrogen isotope fractionation, but here only a very low carbon isotope fractionation. We are not absolutely sure what this is, which is illustrating also this is not a golden bullet, and it could give not always conclusive results, but it is possible that you rank or characterize your data. Yeah? And this is an example from a very contaminated clear with a very complex field. With this, I want to thank you for your attention. So other concepts, for example, biomarkers for activity or metabolites, it's molecular biological tools or backtrack. Here my colleague Carsten Vogt will talk about that. So I thank you for your attention, and if you have questions, we have now also some minutes here to talk about the opportunities of the isotope fractionation concept to characterize microbial degradation. Thank you very much.